So today we'll start by talking a little bit more about genomic alignments. And just to recapitulate from last week, what we were looking into was after you have your sequencing done, your result are reads, which you now know are contained in FASTQ files. And we need to figure out where exactly in the genome they came from. So that's the mapping step we'll go over today. And now you know also that the reference genome for the mapping is contained in FASTA files. So you have all the bases to get this process started. All right, so after the mapping, we can generate the coverage profiles and start to interpret this information. So we'll go through the analysis that we mentioned for RNA-seq and for genome alignments and go into some details. All right, so first of all, let's define mapping and alignment. Mapping is the process of finding the region or regions that read sequence can be aligned to. So there can be more than one position in the genome where a read will be mapped to. And alignment is the detailed placement of each base in that read. So the optimal base by base alignment of the read sequence to your reference sequence. So for each mapped location, you can examine the alignment, for example, and observe if there are any mismatches, if there are gaps, if there are insertions, or if it aligned perfectly to the genome, which is not usually the case. All right, but what are the challenges? The challenges are that the genome, like we said before, is large. And as you can see here, it is composed by a lot of repetitive sequence. So I took this view from Repeat Masker. It is a database of repeated elements in the genome. And here, the black portion of this pie chart is showing the genome fraction that is not composed of repeats. And you can see that it's smaller than 50% of the genome. So that means that all the rest are different kinds of repeat elements that are spread throughout the genome. And that makes alignment really hard. So just another analogy to go through this, it's like we in the Garber lab, we love assembling puzzles. And I had a really hard time the other day that I was trying to assemble one that was composed of snow. So this is just to illustrate, it's kind of similar to when we try to map something to the genome. You have this puzzle piece, this puzzle image, which is basically composed of 4 colors, blue, white, and so on. And you're trying to find a unique pixel in this image that should be the size of a building, and to be an accurate reference to the genome. And it's really hard because you have multiple positions where you have that exact same color that's gonna, that pixel is gonna be a good match to. All right, so let's go through what do aligners do to be able to go through the large genome and to find the best match and not take a week to do so for all your 15 million reads that you generated in your sequencing run. So the answer to that is that aligners create indices. So what is an index? It's just like the index for a book. So it allows you to quickly find sequences that are contained in it. So we'll go through this example for a hash-based index, and we'll see if it becomes more clear. Let's say we have a toy genome. In this case, instead of 3 billion bases, we're only going to have 16, and this is your entire genome. If we were to create an index for this genome, we first have to choose what is the hash size, so what is the k-mer size for this index, and we chose 3. That means that we're going to create an index that is composed of 3 letter words. So the first word in this genome will be CAT. And we annotate in the index what are the genomic positions where we see this particular word. In this case, we only see it here, and we don't see it anywhere. Well, the first time we see it is in position 1, and we just walk over one base and continue writing down the different words that we have. So the second word we find is ATG. We annotate it as position 2, and we go on moving one base at a time until we get to the end of this genome. But as you can see, whenever you reach a word that is repeated, so for example, we see a second occurrence of CAT here, you're also going to annotate it in the same position you had for the first one. So CAT can be found in positions 1 and position 7 of the genome. So this is a complete hash index of our toy genome. And now we can see how easier it's going to be to map some read into it. All right, so let's try some hash-based mapping. So we're going to use the index to find the alignment location for this read. So we got a sequencing run result, and our read is 6 bases long. It's this one here. We want to know where it matches the genome. All right, so first of all, we split the read into the same sized words as we used to construct the hash index. So the first word in our read is TGG. And now we look for in our index, where can we find a match? And we find a match here, and we write down the positions that possibly this read could be in. So based on TGG alone, this read could be starting in position 3 or position 10. And then we find TCA. So TCA is in position 6. We write that down here. But now that eliminates some possibilities. For example, with the combination of TGG and TCA, it's much more likely that this read came from positions 3 and 6, because that would make it contiguous, as opposed to it having a gap and being aligned somewhere else. So we found the correct mapping of this genome really easily by breaking it down, instead of having to match this read to every possible location it could have come from in that genome. All right, so what is a good choice of hash size? So in this case, we had a small genome example. We used a small word. But if you think about the human genome and you think about a K size of 10, so words of 10 bases each, you can have up to 1 million different words composed of 4 nucleotides that are 10 bases long. And every one of these words is present in the human genome at least once. So even with a k-mer of size 10, you will certainly find many different positions in the genome where a similar word came from. All right, so what I want to show with this graph is that it takes a long k-mer to be unique in most genomes. So what I'm showing here, for example, you can see this start around 10 bases. This line here represents 50 bases. And if we follow it up, you can see that with 50 bases even, you're not 100% guaranteed to find a unique position for your read in the human genome. The human genome is shown in blue. And with 50 bases, you would get about 92.5% of your words to have a unique position, but not all of them. So think about this in terms of the sequencing reads that we usually have. 50 bases is a very regular size we have. Not all of them are going to match uniquely to the genome. That's already to be expected. And if you do smaller sequencing where you have reads of 30 base pairs or even smaller, it's going to be even harder to find that unique mapping position. And some genomes are just weird. For example, this parasite Trichomonas, you can see that it follows a completely different pattern than all the other genomes shown here, where it would take a word of 800 bases to make it to make all of them unique mapping positions to their genome. So that's how repetitive it is compared to the human genome. And it's actually a very small genome too. So that's really curious. All right, so we talked about hash-based alignments, but that's not the only way to do an alignment, to create an index, I'm sorry. So there are other ways to create indices, and some of them are very commonly used in mapping algorithms. So for example, we have this strategy called space seeds, where there are 4-letter words that are masked, and then masked 4 letters, and then again a 4-letter word, and all of this is stored in an index. And you first try to match the words that are not masked, and then later on a second step you match the words that are masked. So this is a good strategy for alignment, but it takes tens of gigabytes to store the entire human genome in this sort of seed index format. So it's not very memory efficient, and it actually consumes a lot of resources to do so. So a strategy that has been used with success and initially came from algorithms that are used to compress files is called the Burrows-Wheeler transform. So for example, Bowtie is based on the Burrows-Wheeler transform and is one of the mapping algorithms that we'll use the most. If you're curious about how it works, I suggest just giving a quick Google to the Burrows-Wheeler transform. I'm not going to go into the details, but it's basically you take a string. So in this case, your string is the entire human genome, and you create all possible combinations of it by switching one base, by rotating it one base. And after you do that, so you concatenated the whole human genome into one string, you created all possible iterations of it, the Burrows-Wheeler transform will just take you order them lexicographically, and the Burrows-Wheeler transform will take just the last letter of each of these and form a single string that it can use to reverse match and find that position in the genome. So it works in a really fast way, and the index is only 2 gigabits large. So it's about the same size as the genome unindexed, and it still serves its purpose for quick searches. All right, so then again, after we map, we still have some uncertainty, and we quantify this uncertainty about where this read alignment came from with the mapping quality. So it's the same as the probability that we used for base scores before to evaluate if a base was a correct call or a wrong call by the sequencing software. But now it's going to represent the probability that the sequence should be mapped there and only there in that position of the genome. So mapping quality also uses a Phred log scale, which we learned for the base qualities. So again, the formula is -10 multiplied by the log of base 10 of the probability that you got the mapping location wrong. So in this case, if you have the probability of getting the mapping location wrong as 0.1, then that means that log 10 of that would be -1, and the mapping quality would be 10. So with a mapping quality of 10, it still means that you could have gotten it wrong. You have a 10% chance of having gotten that alignment wrong. So this is what we use to interpret the alignment in the end of the software. And looking at the distribution of mapping qualities could be interesting for troubleshooting, for example. You have a mapping run where 80% of your reads align, you're probably satisfied with that. But if you have smaller and smaller fractions of your reads aligning, then you're probably going to want to delve into this and see what's happening to my reads. Is there a problem with mapping qualities? Does this mean that maybe my sequencing library is not good and I should re-examine the quality of them? All right, so let's just recap what we went through. So the read alignment relies on this dictionary we created. So once it's loaded into memory, and now you see the efficiency of an index that is 2 gigabytes large, or an index that is 20 to 40 gigabytes large, you can process thousands of reads per second. And different aligners are going to offer different advantages. For example, we have a variance of sensitivity and variability, so you can align to highly variable organisms. It might require you to have different parameters or even a different aligner to begin with. You have differences in speed, so some aligners like HiSat and Bowtie is actually super fast, are going to offer different advantages in terms of speed compared to others that are super slow but maybe more accurate. So it depends on what you're looking for. Memory efficiency based on the indices mostly. Specificity, which comes at the cost of the sensitivity of your alignment. And this is by no means a solved problem. There's always new alignment algorithms showing up. So you shouldn't assume that the one you're using now is the same one you're going to be using 5 years from now, although some of them have become quite staples in our arsenal for pipelines, and they are still some of the best to solve this problem. And in the end, I want you to keep in mind that you really don't know which region was actually sequenced. All you know in the end of this process is what is the best match of your read to the reference. So that is the best guess you can make, and it's good for most applications, but it's not a guarantee that you'll never know which place in the genome your read actually came from with absolute certainty. So one of the factors that leads to this is that there could be sequencing errors. So your reads are not the exact match to the same fragment that they were created from, because the process of sequencing them introduces errors to begin with. And you also have variability from the genomes itself. So you're aligning to a reference genome, and you have to think that that genome is not going to match the genome, the exact genome of the cell you're growing in the lab. So there's been several different mutations that have happened in the lab. And other than that, there's variability from human to begin with. So the human reference genome is a composite of several different humans, but it is to be expected to have several different mismatches to the reference genome, and that will impact alignment too. And lastly, so you will have non-unique alignments. That means you have positions in the genome that are repetitive, and therefore you will have multiple possibilities where that read came from originally. All right, so this is just to show you the abundance of sequencing aligners that are out there. This is a table from 2015, and it's just listing all the algorithms, and you have here what kind of properties that algorithm has. And this is just a piece of the table. It actually goes on for longer and longer, and it doesn't even contain all of the algorithms that are currently available, and they're actually used for a lot of different purposes. So let's just summarize that to the ones that we currently apply to more to our data sets. You have aligners that are specific for DNA-based reads. So reads that came from the genome to begin with, like ChIP-seq reads, ATAC-seq reads. You're probably going to use Bowtie, which is the one that uses the Burrows-Wheeler transform index, like I mentioned. And it's good for finding mapping for single-end reads as well as paired-end reads, and even for finding structural variations if you have them in your data and if you have interest in finding them. And in terms of aligners that are good for RNA-seq reads, and I'll show you in a minute why they're different, you're going to use STAR today to align reads to the genome. And it creates an index that is hash-based. So it's like the example that I went through line by line, that creates an index like a book that tells you where each word is found in the genome. And let's go through why it's different to have aligners that are made for RNA-seq compared to DNA reads. So what is the RNA-seq mapping problem? RNA-seq comes from a large majority of mature transcripts, right? Which means that for genomic mapping, we need to consider that this transcript, where they originated from, has been spliced. So it's not going to be found in the genome exactly like this. So many reads will likely span more than one exon, and therefore we cannot align it to the genome in a single stretch. So how do we do that? So we have FASTA files that contain the sequence of the genome, and we have the annotation files that contain the positions of known exons of known transcripts in the genome. So an aligner that is hash-based can try to, for example, star, can still map these reads because it first tries to map the biggest stretch that it can from a read. And when it can't align anymore, it does a second alignment separately for the remaining stretch of that read. And in the end, you're going to have a correct alignment for your reads that are entirely contained in exons, as well as the reads that span more than one exon in the genome. And another possibility is that you can have an aligner that is not splicing aware. So this is what we call a splicing aware alignment. And you can perform that alignment using a transcriptome reference. So this is something you're going to do today too when you use the RSEM program for RNA quantification. That program is actually in the backbone using the bow tie aligner, and it's mapping your reads to a reference transcriptome. So what is a reference transcriptome? So just like the genome can be contained in a FASTA file, so can the sequence of each of the known transcripts. So this is a already spliced, mature sequence of each transcript you know you have in your genome, and it can be used for mapping the reads directly onto it. So now there's no problem anymore with reads that will map to more than one exon because the exons have been spliced computationally. All right, and the advantages to this process of mapping to the transcriptome, for example, are speed. So think about if the transcripts are about 2% of the genome, that means that you're aligning to a much smaller universe of probabilities. And therefore, the alignment will go much faster. You don't have to look for in the entire genome for a probable match to your sequence. But what are the disadvantages? The disadvantages is that it relies on having a quality transcriptome annotation. So if you're working with an organism that is not widely used, you probably don't have that available to begin with. And you're more likely to be stuck using the genome and then making inferences of where your transcripts came from. But there's also another issue, even for genomes where you do have a good reference annotation, and that is you will not discover potential new isoforms. So if the goal of your experiment is to find novel transcripts and novel isoforms, that cannot be done if you only inform your aligner of the transcripts you know. So mapping to the genome will maybe give you a new isoform that doesn't contain an exon, for example, or that contains an exon that was not known in this in the particular annotations that you have now. So it all depends on your goal. And then lastly, let's just go through this strategy again that these aligners do for mapping first the exon first spliced alignment. So softwares like Top Hat will first try to map all of the reads that it can that are contained wholly inside exons. And it knows where the exons are because, like I said, we feed it not only the genome FASTA sequence, but we also feed it a GTF annotation file that tells it which positions of the genome contain an exon. So it'll first map all the easy reads, let's call them. And then later, it'll do splice read mapping, where it knows all the junctions that should be there, and it even is able to find new junctions that you didn't know were there. And it maps part of these reads only, and it can stitch them together in the end and find the splice read mapping. All right, so I'll leave you guys with some references. You probably noticed that I used a lot of the slides, including the toy genome from Aaron Quinlan. And there's a lot more from his lab that can be found here in this link. And if you want to go deeper into the bioinformatics aspect of some of these problems that I talked about today, that's a good place to look for some videos even. And if you want to read more on the problem of mapping billions of short reads, this is also a really good paper, and although it's from 2009, it has aged very well. And this is the reference for the repeat masker. And now you guys will go through the genome alignment exercise, and we'll come back for transcriptome quantification.